Hello, ladies and gentlemen. I'm Daniel Montano. I'm the Chief Executive Officer of Zhittya Genesis Medicine. First, I want to thank you all for being on this webinar for our presentation on can human FGF1 be a potential treatment for ALS. I'm going to introduce our topic, and Dr. Jacobs, my partner, who you can see on the screen there, will be giving you the primary presentation. But I think it's very important to give you some background on the situation. First of all, Zhittya is the Ukrainian word for life. Zhittya Genesis Medicine means life's new beginning. And we're going to be talking about our molecule FGF1. Jack and I have been on this journey of discovery for over 20 years now. We have expended in excess of $140 million, and we presently are working on 19 medical indications, from heart disease to peripheral artery disease, and then diseases in the brain, such as Parkinson's, and obviously ALS, topic of today. I'd like to make sure that you understand that the whole purpose for us to have these webinars is to make you aware of what we're doing. We are not selling a product, we are not selling a service, and we are not seeking donations. We are a biotech pharmaceutical development company. Until we get regulatory approval, we have no products, no services we can sell, and we take no donations. We are here to explain to you and make you aware, because when it comes to clinical trials, the number one reason clinical trials fail is lack of volunteers. According to printed reports, 70% of all clinical trials fail for lack of volunteers. Equally so, another report states that 70% of the people who have a disease that want to be in a clinical trial cannot find out how to get into one. So this is what we are working upon, is making you aware. Now, one of the things that people have come to us and asked, why are we working on this, ALS? First of all, Dr. Jacobs and I started about 4 years ago working on Parkinson's disease. And Jack will use some references on our Parkinson's progress because we believe that Parkinson's and ALS are related cousins, and he'll explain that to you. But about 2.5 years ago, I got a call from a friend of mine who is a money manager in Bangkok, Thailand. His name's Philip Wilson. And Philip and I have been friends for 30 years. Philip is a money manager. He manages a couple of $2 billion in investor money, and he has invested with me when I used to be an investment banker in London. And he and I are good friends. Philip called me up and said he had read the work we're doing on Parkinson's, and that I think it would apply to MND. And I told him I have no idea what MND is. And he said, Dan, motor neuron disease. And I said, sorry, I still have no idea what that is. And he said, oh, it's the British terminology for your ALS or Lou Gehrig's disease. So he informed me that he had just been diagnosed with it. I called Dr. Jacobs. Dr. Jacobs did some research with our research team and came back and said he thought there were parallels with Parkinson's and that there was a chance we could address it. We have been going as fast as we can to find out if our molecule is safe in the brain, and two, if it can change or modify the direction of ALS. We have filed an application in Mexico, and we have been approved in Mexico to do an ALS clinical trial. But what I want to give reference to is when we filed the application in Mexico. Now, please understand, we filed an application in the US for Parkinson's, FDA, IND. The US wanted us to do additional 2 animal trials, which we are doing. However, we were informed that Mexico would accept the animal data we had. We thought for Parkinson's we should try. We submitted the application, and Dr. Jacobs said we should submit an application for ALS and get that clock going, because we didn't have time to wait to find out the outcome of the regulatory authorities in Mexico. Months matter to ALS sufferers. So we filed that application. We just got approval. Unfortunately, the COVID-19 has delayed many things, but we are making progress. And I want to emphasize, we are here to explain the medical science. We are here to explain why we think our molecule might be able to change the course of this horrible disease. I'm going to introduce my partner, Dr. Jack Jacobs. Jack has been my partner for 20 years. He has a PhD from the University of Washington in Missouri. They refer St. Louis, Missouri. They refer to as WashU. Jack has a PhD in molecular chemistry and biology. He worked at Hitachi Pharmaceutical. He went to Merck, where he set up their biologic manufacturing facility. Jack has been working with this molecule since it was first identified in the 1970s, which is very impressive that Jack's been working on this for over 50 years and he's only 45. Jack is, besides one of the smartest people you'll ever meet, he is one of the kindest hearts and one of the most nice people you'll ever meet. Jack takes this project very serious. We understand that this is something that we got to find out now if it's real. Dr. Jacobs, please take on the presentation.
Okay, thank you, Dan, for that very nice introduction. So as Dan mentioned, I'm going to be drawing some parallels to some other work we've done in the heart with Parkinson's disease. And I believe this directly applies to potential treatment for ALS. I'm going to talk a little bit about our drug and how it works. It grows new blood vessels in our bodies. It's probably one of the most potent growth factors to stimulate new blood vessel growth in our body. So we're using it in diseases that are characterized by a lack of blood flow. And we think Parkinson's and ALS are initiated by one route is by lack of blood flow. Here's an emerging medical hypothesis held by us and also other researchers. They believe that inadequate blood flow in the brainstem and spinal cord initiates ALS, leading to motor neuron death. And through my talk, I'm going to be showing you why we believe a lack of blood flow, coupled with other things, hereditary, environmental factors, can initiate and lead to the progression of ALS, as well as other neurodegenerative diseases. So here's the target in ALS is the motor neurons, as you all know. These are very large, long, and very hungry neurons. The motor neurons, in fact, are 5,000 times larger than the typical cell in our body. They can often extend their axons great distances in our body to reach target muscles. So these are the neurons that are affected in ALS. And in fact, the longest neuron in the body, the sciatic motor neuron, is over a meter long, stretching from the spinal cord all the way down to the toes. And these motor neurons have incredible energy needs. To transport a protein or a macromolecule, this entire length of the leg takes a lot of energy. So these cells, we believe, and others, are exquisitely sensitive to a decrease in blood flow, a decrease in something that would prevent oxygen, glucose, to get to these neurons which have high metabolic activity. And remember, they're electrically firing all the time. So we believe this lack of blood flow, which occurs as we age or can occur with trauma, say with a traumatic head injury, overlaid with genetic factors, environmental factors, is the initiating cause of ALS. I'm going to talk now a little bit about this process of our drug growing new blood vessels. It's a process called angiogenesis. We use it all the time. So I'll talk about natural angiogenesis, how we use it, and also how we've used it in FDA clinical trials to grow new blood vessels in the heart. I'll show you some data from Parkinson's a monkey model. We have some data available for the ALS mice models, and then I'll talk about our clinical trial, which we hope to be starting very soon. So we all have 60,000, at least 60,000 miles of blood vessels in our bodies. These are all formed by the process of angiogenesis, which is the growth of new blood vessels. And we know if we get blockages, let's say in our coronary arteries, we can develop heart disease. Blockages in the arteries leading to the brain can lead to stroke. And as I mentioned, newer research indicates that Parkinson's disease and ALS can be initiated by a lack of blood flow to selected areas of the brain and spinal cord, basically choking off with Parkinson's, the dopamine neurons, and with ALS, the motor neurons. So angiogenesis is shown here. You can see all these new blood vessels which form generally in damaged or ischemic tissues. And in fact, over 75 human diseases are a result of impaired blood flow or lack of blood flow to a tissue or an organ. We're working on 19 of these, and I'm not going to talk about all 19, but probably the easiest way to stimulate better blood perfusion into the brain, into the heart, is by stimulating the growth of new blood vessels. Again, a process called angiogenesis. And that is what our drug does. It's a natural product, a natural growth factor in our body. It's probably the most potent stimulator of new blood vessel growth. And we use it all the time. You use it all the time. We cut ourselves. Here's a skateboarder falling off a skateboard. He's going to use angiogenesis to heal this wound. So let's take a look at this. This is the process of natural angiogenesis, and we differentiate that from our therapy, which we call therapeutic angiogenesis. But naturally, let's take a closer look at his elbow. Here's natural angiogenesis in this wound. This is, let's say, the scab that's on his elbow. Let's look underneath that scab. Here we see all these new blood vessels, which have formed in response to his injury and in response to FGF1, the drug we are developing. We see these new blood vessels not only in these wounds, but I'm going to show you, we see them in the heart, we see them in the brain, and these new blood vessels bring nutrients to regenerate skin, and skin, not just skin cells, but everything needed for skin to be a whole organ: sweat glands, hair follicles, nerves, new blood vessels. So this regenerates skin in this wound bed, and I'm going to show you how we can regenerate neurons in the brains of animals with Parkinson's disease and also with stroke. Okay, here's FGF1. Dan mentioned I've been working on this, I don't think for 50 years, Dan, but maybe a good number of years. It is actually first purified at Washington University. It's a protein, so it's a natural protein product, like growth hormone or insulin, obviously not the same structure. And we're using a recombinant form, which we manufacture in an FDA-inspected facility. It's human FGF1, which we'll be using in our clinical trials. So let's look at how FGF1 stimulates angiogenesis. So acutely, when that skateboarder wounded his elbow, immediately his body started synthesizing, producing FGF1, shown as these little flying guys. And they seek out blood vessels. Here's the FGF1 receptor. So it's like a key going into a lock. FGF1 binding to its FGF1 receptor, and that stimulates this process of angiogenesis or new blood vessel growth. You get an explosion of metabolic activity. The cells that make up the blood vessels start dividing. Here we see the very beginning of a new blood vessel growing into the wounded area. So it's a directional growth. We see a tremendous upregulation of the receptors. So we only get new blood vessel growth where we need it in the wounded area. And this happens thousands of times along different vessels in the wounded area. We call this a foci of new blood vessel growth. And that's going to bring in all the nutrients and extra nourishment that is needed to heal that wounded area. Now, in chronic diseases, so this is not acute diseases like the wound that skateboarder got, but this is chronic diseases, heart disease, Parkinson's disease, we believe ALS. The tissues become ischemic due to a lack of blood flow. There's a choking off of the blood supply. And the regions of the brain or the heart cry out with upregulating their FGF receptors, as shown here schematically. The problem is, over time, if it's chronic, if this is happening over years or decades, for some reason, there's not enough FGF1 around naturally to form new blood vessels to try to give the blood perfusion a boost in those ischemic tissues. So that's where we come along with therapeutic angiogenesis. We actually inject, this is the yellow ball, FGF1, injected into the heart, injected intravenously and getting into the brain. This is going to stimulate this whole process of angiogenesis, new blood vessel growth, getting new nutrients into damaged tissues. And I'll show you how this is coupled to the production of new neurons in the brains of animals. Let me first show you some work that I supervised and did. This is FDA supervised, FDA sanctioned trials, which I supervised, injecting FGF1 directly into the hearts of patients with severe coronary artery disease. And I'm showing you this just to emphasize that we can grow new blood vessels in the human body, in this case in the heart, and we feel we can do the same thing in the brains of patients. So let me show you, actually ABC News came down to our University of Cincinnati clinical trial site and interviewed the main cardiologist, Dr. Lynn Wagner, and also some of the patients who came into the trial. These were mainly cardiac cripples. They had no other options. They couldn't have a bypass or a stent. And I want you to watch this video that ABC News put out. You're going to see Dr. Wagner pointing to the area of the heart which was injected with our drug, FGF1, and you're going to see new blood vessels growing there about 3 months after the therapy was introduced. So I'm not going to talk over this video. I'll come back at the end, but please watch it.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
So that woman we saw there, Constance Donnelly, she came into this cardiac trial in a wheelchair. And you can see her there 3 months later, up and about. And her heart must have been highly upregulated in the FGF1 receptors in her heart because she responded very well. She was actually in the lower dosing group in this trial. So responded very well. And we're going to be moving ahead with our heart trials as well. Let's move to the brain now. The brain is probably the most vascularized organ in our body. We know we have billions of neurons in our brain, and they need billions of capillaries and small arteries to supply them with nutrients, oxygen, glucose, and also importantly to remove metabolic waste products that all cells in the brain generate. Here's an important concept, a neurovascular unit. This is where the blood supply in the brain interacts with neurons. We go from a large artery down to a smaller arterial, to a capillary. So here's a fully functioning capillary. I'm going to show you later on, we know capillary density is down in animals with ALS, densities decrease in the spinal cord. Here in a healthy brain tissue, we have a fully open capillary, which feeds these adjacent neurons. So blood flow supplies this neuron with glucose, oxygen, removes waste products. And you can imagine if this capillary became blocked with atherosclerosis or was damaged by inflammation or environmental factors, these adjacent neurons are not going to be doing as well. Also, what these capillaries perfuse, and not shown on this, are stem cells. We all have neural stem cells, which are in our brains in reserve to grow and differentiate into mature neurons, mature motor neurons, if needed. But we'll see that a lack of blood flow in certain diseases, and as we age, makes these stem cells less active. And we also think that's contribute to these neurodegenerative diseases. I'm going to show you now some evidence we have that FGF1 can grow new blood vessels in the brain. We're going to look, this is in a stroke model. And again, this is important because it shows that we can give our drug intravenously. It can cross the blood-brain barrier and stimulate new blood vessel growth in the damaged brain. And we think this is directly applicable to both Parkinson's and ALS. We used a mouse and a rat model. We can tie off an artery leading to the brain. This is not a mouse brain, this is a human brain, but in a similar way, the mice develop a stroke. And this is an area of dead tissue and at-risk tissue. So these are neurons which we can save, and we can try to regenerate neurons with our drug FGF1 in this model. And in fact, that's what we did. Here are slices through the brain of rats, and we also did mice. You can see this is the infarcted area. This is the stroke vial. This is the area we're going to try to treat. These are the FGF1 treated animals for 2 weeks. You can see clearly we are decreasing the volume of that stroke. And we can look in this area. We can look, are we generating new blood vessels? Is angiogenesis occurring? Is that linked to neurogenesis or new neuron growth? And in fact, it is. So let's look at the blood vessel growth. Here's capillaries in the mouse brain. Not shown here are the millions of neurons which would be fed by these capillaries. We tie off an artery leading to the brain. We induce a stroke in these mice, and you can see the decimation of the capillaries in those animals. These animals got FGF1. 14 days later, we see a nice regrowth of capillaries in the damaged area of the brain. Not quite back to normal, but certainly better than the placebo-treated animals. Here, the damaged brain is desperately trying to regrow blood vessels. It grows them in a disordered way. This clumping can lead to more hemorrhaging in the brain. These animals do worse. And we can infer that they're growing new neurons because we can do some mobility testing. This is in mice that received a stroke. Normal animals this is a rotating rod. They kind of hang on well. Here's the FGF1 treated animals, not quite as well, but they're hanging in there. And then the placebo animals basically fell off that rotating bar. So we see in this animal model that FGF1 can grow new blood vessels. That's coupled to new neuron growth, and the motor skills in these mice improve. Now let me show you some data in primates in a monkey model where we can regenerate neurons in the brain. And again, this is going to be in a monkey model of Parkinson's disease. And this is what we use to justify going into humans. And as Dan mentioned, we're set to start a human trial in Mexico in Parkinson's disease. So the gold standard in testing drugs in Parkinson's is a primate model where you can give these monkeys Parkinson's disease. You can inject a toxin. It's actually a herbicide related to paraquat into the brains of these animals. And after a month or so, or a few months, they come down with irreversible Parkinson's. And we can treat those animals with our FGF1 and see if we can improve motor skills. And also, are we regenerating the target neurons? In this case, it's the dopamine producing neurons that cause Parkinson's disease. So here is the results of a monkey test. Monkeys were given this toxin. These are motor scores. So here's normal motor movement in the monkeys. This is highly abnormal movement. We inject that toxin that I talked about, and then they slowly develop all the classical symptoms of Parkinson's disease. So at this point, these animals have tremors, they have gait disturbances. Half receive an infusion of FGF1, the other half continue to receive placebo dose. And at the end of the experiment, we can see a very significant difference in the motor skills between the FGF1 treated animals and the placebo treated animals. These animals actually are close back to normal. If we perhaps treated longer, we could have gotten them back closer to normal. But what we wanted to see in this study was, are we regenerating new neurons in these animals? And is that regeneration coupled to this improvement in their motor scores? And indeed we did. Here you can see the area of the brain. It's called the substantia nigra. It's an area very deep within the midbrain of the monkeys. This is where the dopamine cells are that become dysfunctional in Parkinson's. You can see here the new dopamine cells stain brown. We are regenerating new dopamine cells in those monkeys. Placebo, no new dopamine cells. And we believe this regeneration of dopamine cells is attacking the root cause of Parkinson's in these primates and leading to their better motor scores. And that's why we're so excited to get this into the clinic in patients with Parkinson's. Now with ALS, FGF1 has been looked at. This is not our work. I'm going to refer to some academic work. But the main model for ALS is ALS mice. They have a mutated gene. It's a superoxide dismutase #1 gene. So this is a free radical scavenger. And in fact, in humans, 5 to 10% of ALS is hereditary. And the majority of hereditary cases are due to a defect in this gene. So these animals have this defect and they develop ALS. They develop paralysis, and generally within 140 days, they die. So most drugs that are tested before the clinic go into these mice. So let me show you some data. This is coming from the Maine Medical Center in Portland, Maine, that tested FGF1 in these mice. Let me just show you here the spinal cords of these mice. These are the capillaries, normal mice, higher density of capillaries than in these ALS mice, where you can see lower density of capillaries. So testing of FGF1, we're going to look at two parameters, time to paralysis in the mice and also survival. There's some important qualifications that I want to talk about in these animal trials, but let me show you the data first. So half the group gets saline in red, the other half get FGF1. And you can see a signal here. This is not obviously reversing the disease, but this is a signal between saline and FGF1, which we believe is worth exploring. These animals have delayed paralysis. This is the days where their paralysis comes on, and there's clearly a difference between the FGF1 treated animals and the saline treated animals. With survival, again, we see the same thing. FGF1 is in blue. We start to see a separation of the two groups as we go out in time in terms of survival. Now, the qualifications that I want to mention here are two. One, the dose of FGF1 given is extremely tiny. It's a microgram. So in our clinical trial, which I'll talk about in a second, we're going to be given 10 times that dose, 10 micrograms. The second thing, and this is very important, the drug in these mice was given subcutaneously, which means it was given under the skin. And having worked with FGF1 for 30 years, I can tell you, if you give a small dose under the skin, not much of that is going to get to the brains of these animals. So we're going to be giving, in our clinical trial, the FGF1 at higher doses intravenously. Actually, we're going to be giving it into a central line, so the FGF1 will get right to the heart, and we believe quickly to the brain. So we believe hopefully we can exploit this difference and see a much better effect. Now, work from a second academic group looked at the ability of FGF1 to induce motor neuron regeneration in ALS mice. And they looked at, this is the lumbar 1 motor neurons. So these are neurons kind of in the lower spinal cord of the mice. They look for growth, regrowth of those neurons. So here's normal neurons. In the normal mice, ALS mice obviously have a decreased number of motor neurons in that region. And then those mice treated with FGF1 regenerate, obviously not back to normal, but a significant amount of new motor neurons can be generated by giving these mice FGF1. So we think this is good data, important enough data to convince the Mexican authorities that that FGF1 was an attractive candidate to perhaps treat ALS in humans. And I'll talk about the clinical trial, which we'll be doing down there just in a second. I want to allude to one other pathology that we believe can also be involved in these neurodegenerative diseases that can cause a lack of blood flow. And this is vessel deterioration as we age or through trauma. And we believe this may be playing a part in Parkinson's as well as ALS. That is, our vessels get leaky as we age or in these neurodegenerative diseases such as ALS. And we're going to look at these endothelial cells. They line each and every one of our blood vessels. And newer work has shown that these can become leaky, and they can leak toxic elements out into the surrounding neurons, which can make those neurons dysfunctional and injure the neurons. This is called primary endothelial cell dysfunction. This is observed in, again, capillaries. These are the normal endothelial cells lining all of our blood vessels. You can see they're tightly packed, kind of like a nice tile floor. Nothing is leaked out. And then there's this primary endothelial cell dysfunction. You see the endothelial cells become malformed, bumpy, leaky. And it's been shown in multiple sclerosis that this can lead to damage of the surrounding neurons. And we think the same thing is occurring in Parkinson's, ALS, and I'm going to show you a video where this also occurs as we get older. And we know that the average age of onset for ALS is between 50 and 60 years of age. So here's conceptually what we and others believe is going on as the blood vessels become dysfunctional. Here's a healthy capillary, optimal blood flow, healthy neuron. And then this is in a model of MS. No one really knows what causes multiple sclerosis, but genes, smoking, lack of vitamin D, pathogens. But the vessels become leaky. And they're leaking things out that shouldn't be outside of the blood vessel cell. They're leaking out immune cells. They're leaking out fibrinogen, which is a protein, a very sticky protein that normally is involved in blood clotting. It has no role being outside of the blood vessel system, but that can coat these neurons, make them look foreign, cause more immune attack. And this whole cycle just damages neurons. And again, we think this process is occurring as we age and also in various neurodegenerative diseases. FGF1, our drug, can heal this situation. Let me show you some data where FGF1 can restore and heal these damaged endothelial cells which line the capillaries. Again, this is in an animal model of MS, multiple sclerosis, where we know the vessels are damaged and leaking toxic things out. You see a vessel in a lesion from an animal with MS. And over about a 30-day period, we can give FGF1 intravenously, and we can see a very nice healing out here at day 30, a complete healing of these vessels that suffer from this dysfunction, endothelial cell dysfunction. These vessels are no longer leaking the toxic substances, and healing can occur. Healing of the neurons can occur in these animals. We can look at the vessels histologically, stain them. These are the endothelial cells at day 10 in that model, leaky still, healing, and then fully healed endothelial cells induced by our drug, FGF1. So if vascular dysfunction initiates ALS, and we and others believe this, then we strongly believe that FGF1 is an excellent candidate to test in the clinic. We know in animals that between 1 to 5% of the dose which we give intravenously can enter the brain and spinal cord. And once in there, we believe it'll drive these four main natural processes. And I've shown you data for this in animals, in humans. Angiogenesis, the growth of new blood vessels, we saw that in the heart. We saw that in the brains of the rats that had stroke. Neurogenesis from stem cells, we saw that in the Parkinson's monkeys. Neuroprotection was seen in the stroke model. And then I just showed you healing of endothelial cell dysfunction in that animal model of MS. We believe all four of these processes can be very important to healing vascular dysfunction in a neurodegenerative disease such as ALS. And this is why we believe this is an excellent candidate to go into the clinic. So here's the phase 1 clinical trial for the ALS trial, which will hopefully start soon in Mexico. We'll be looking at 3 ascending doses of the drug. There'll be no placebo group. So all patients will receive a 1-hour intravenous infusion of FGF1 5 days a week. We'll give the patients a weekend off through a PICC line. This is a central line, which we believe gives the FGF1 the best opportunity to penetrate into the brain and into the spinal cord. This is a proof of concept study. We're going to be looking both at safety and effectiveness. The neurologists running this trial will be using a revised ALS questionnaire, and we'll follow the patients for a year. So this is primarily a safety study, but we'll be looking at efficacy. As long as our FGF1 is safe, we can go on and increase the dose we give to ALS patients. We can give it for longer periods of time. So this is our first proof of concept trial. And I want to talk a little bit about Mexico. I've gotten questions, are you doing this in a, stem cell clinic across the border in Tijuana? And definitely not. We're doing this at a first-class hospital, one of the best hospitals in Latin America, Hospital Zambrano in Monterrey. It's new, it's superbly equipped, great staff, and we feel really confident that this will be an excellent clinical study. Importantly, it's going to be done to FDA standards. We've hired a clinical research organization, Cohortius, based in Europe, but they have a branch in Monterrey, so they know the staff at the Zambrano Hospital very well. Trial will be run to good clinical practice standards. We have a wonderful, highly trained neurologist, Dr. Martin is down there. He's trained at the National Institutes of Health as a movement disorder neurologist. And I want to emphasize, Dan mentioned this, that we're going to pay all the costs of the clinical trial, including travel and housing expenses for people who come from outside of Monterrey or outside of the country. And we're seeking no donations to carry forth with this trial. And we hope to start this as soon as possible. As we all know, time is of the essence for patients with ALS. I'm going to close my talk. I'm going to show you a short video, and this ties into all that we've been talking about how blood vessel perfusion of the brain is important in these diseases. I'm going to show you some work from Columbia University Medical School, a very prestigious medical school in New York City. They published this work in Cell, a very prestigious journal. And they show that lack of blood flow to the brain actually makes our brains perform worse as we age. So they're going to be looking at normal people. I'm going to give you the punchline of the study before I show you the video. Basically what these Columbia researchers are going to show that we need adequate blood perfusion to stimulate neural stem cells to divide and mature. They're going to show this in normal people, young people versus aging people, and why as we age, our brains, the memory part of our brains, perform less well. We believe the same thing is occurring in Parkinson's and ALS, that lack of blood flow causes the stem cells to be less active. So in ALS, less active, less differentiation into motor neurons, and Parkinson's less differentiated into dopamine cells. So let's take a look at this. I'm not going to talk through the video. There's text that occurs through the video. Then I'll come back to see you at the very end. So we agree completely with this video. We believe blood flow, blood perfusion to the brain is very important as we age and also in these neurodegenerative diseases. And we believe we have a molecule that can restore blood flow in these situations. So thank you for your attention. I'm going to call Dan back now. We want to take any questions. We do have a website, zgm.care. You can go there. You can sign up for the ALS clinical trial. You can request our white paper that we've written on ALS. You can learn about the other diseases, what we're addressing. And Dan, our wonderful CEO, also takes questions directly to his email address here. And you can email him directly with any medical or business questions you have about the company.
Jack, thank you very much. We do have a lot of questions. But first of all, ladies and gentlemen, if you're interested, we have a booklet which basically goes through the biology and how the drug works, how the medicine works, and a couple of clinical examples. You can email me and we'll send this to you electronically. And as Jack mentioned, we have white papers. This one is on Parkinson's. Jack also wrote one on ALS, but that's a couple of years old now. We keep advancing our knowledge base. The other thing I'd like to mention is I want to emphasize again to everybody who's listening, we don't take donations, we don't seek donations, we have nothing to sell anybody. This is a research project. If it's successful and our drug gets approved, then it will be on the market, and that's where we as a company will earn our money. The first question, Jack, came from one of the people. Is the data based upon 1 microgram of FGF1?
So the animal data I showed you for survival and paralysis, yes, was based on 1 microgram actually per kilogram. We do everything by weight. So obviously humans that weigh about 75 kilos, we get a larger dose, but the critical dose is how many micrograms per kilo. And those mice only got 1 microgram per kilo subcutaneously under the skin, where in our clinical trial, we'll be giving up to 10 micrograms per kilogram directly into the bloodstream, directly into the vasculature by the heart. And we think much higher quantities will be available to the brain and brainstem and spinal cord in the clinical trial.
Okay, Jack. Another question from one of the viewers. You mentioned stem cells. Where do the stem cells come from? External?
No, we are not giving, this is not a stem cell trial. So, 20 years ago, people thought we were born with all the neurons we ever were going to have, and that simply is not true. Researchers, first at UCSD in San Diego, and now others, we have pools of stem cells in our brains. As a Columbia University video showed, the pools in the younger people are more active than in the older people. But we all have stem cells, and these stem cells need blood flow to stay active, and they need blood flow to differentiate into mature neurons. So we believe our drug, which is a protein, once in the brain, can reestablish blood flow. In the case of ALS, this leads to regeneration of motor neurons from the stem cells that they already have in the brain. But if they're malnourished, if they're not receiving enough blood flow, they're not going to be active enough to differentiate into new motor neurons, which you need to counteract the symptoms of ALS.
Jack, I'd like you to go back on that same topic and address the slide you showed about the animals that were given a stroke and the dead area, and then the animals that were given a stroke but got FGF1 and how that had repopulate. Why was it able to repopulate?
Right, so those animals, again, got FGF1 intravenously, where they damaged their brains, the stroke area, becomes ischemic. It becomes starved of oxygen due to lack of blood flow. So in that area, the FGF1 receptors on the surviving blood vessels are tremendously upregulated. The FGF1 binds, new blood vessels grow, as we saw, and that's coupled to new neuron growth. So in those stroke animals, they first regrow their new blood vessels, regrow new neurons, and we saw on that motor rod, they're better with their motor skills. So I think that's a very good example of how this drug can function in the damaged brain.
Okay, next question, Jack. Is your drug similar to the Japanese drug that has already exist intravenously, and I think they're talking about radicavia.
Right, radicavia. So radicavia, no, it's not similar. Radicavia scavenges free radicals, and that's thought to be its mechanism of action. From reports I've read, some people benefit from radicavia, but not a huge percentage. So, our drug, we hope, is not just treating a symptom, but it's going to regenerate motor neurons, which is the root cause of ALS. So we saw in the Parkinson's monkeys regeneration of the dopamine neurons. We're hoping that we can do the same thing in ALS patients, regenerate motor neurons, which are the root cause of that disease. And we saw a little bit of that. We saw evidence of that in the ALS mice that were treated with FGF1. They regrew those lumbar motor neurons.
So what I also want to emphasize is that we're not looking at chasing the symptoms. We're looking at addressing the root cause of the disease and thereby reversing the course of the disease and hopefully having the potential for a full recovery. So now another one is, what are the parameters to participate in the FGF1 trials, Jack?
We have, for the trial, we have inclusion and exclusion criteria. Obviously, the neurologist, not myself, will deem whether a person is appropriate, but it's pretty wide open. If you have ALS and can get to the trial, obviously we're going to exclude people who have cancer. If you're female and you're pregnant, you can't get into the trial. But generally, most people with ALS, whether it's early phase, mid phase, can get into the trial. But again, in Mexico, if someone from, say, the U.S. is interested in the trial, once the trial gets up and going, we would ask, electronically, their neurologist could send their medical files, obviously under confidentiality, to our neurologist, our principal investigator. They could be reviewed in Monterey, and the person could be seen whether they could qualify for the trial. But then people would have to be screened in Monterey by our neurologist before they actually could enter the trial. So it is a commitment.
Jack, I'm going to attempt to answer this question. We have a question: why aren't the clinical trials being conducted in the United States? Dr. Jacobs addressed that already, but I'll come back and readdress it to make sure that everybody understands the seriousness of this. We filed an application, an IND, with the US FDA for Parkinson's. When the US Parkinson's disease, when the US FDA responded, they wanted us to do additional animal trials in the United States on 2 species of animals, because this will be the first time FGF has ever gone into the brain. That additional animal trial will take approximately 1 year. We are doing that. In the meantime, we were told that the people in Mexico would accept the animal data we already had. So we submitted an application identical to what we submitted to the FDA to Mexico. And at the same time, Jack decided that we should go ahead and file an ALS one, because if the people in Mexico would accept the animal data for Parkinson's, they should accept it for ALS. We thought time mattered. So instead of waiting an additional year to find out if we can start in the US, we thought we would start dosing people who have ALS in Mexico and find out 2 issues. 1, is the molecule safe in the brain? And 2, do we change their outcome? That's the point. We're not in Mexico because we're not trying in the US. All of our trials, we focus on trying to get US approval. But the US process is extremely slow, and an extra year delay for an ALS patient is a long time. I started this by mentioning my friend Philip Wilson is dying from ALS. We don't have an extra year to watch him. So we went to Mexico because we're trying to find out if this works and if we can treat people. Jack and I take it personal. We're not bureaucrats. We're not pharmaceutical executives. We take the people we meet, we take their lives and their suffering seriously. And a year delay is a long time for an ALS patient. That's why we went to Mexico. Our trials in Mexico may prove that our drug does not work. It may prove that the molecule is not safe. It may prove that we don't change the outcome. If that's the case, we lose money. but we think it's better that we lose money than people lose a year who are struggling to survive ALS. So that's why we're in Mexico. I hope that answered your question. Next question, and Jack, I think you just answered it. Do we accept candidates for the trial? I think you just answered that, right?
Right.
Okay. I got another one here. What are the known side effects?
Well, you know, for the FDA studies, the FDA makes us push the dose of FGF1 we give intravenously. So we went 10 to 100-fold higher than what we're going to give patients. And you have to do this in drug development. We saw some effects on the thyroid gland. One I want to mention, we've never seen cancer induction by our drug. This is a growth factor, so people often ask about that. We've never seen any hint of cancer initiation in either the animals we've treated at very, very high doses or people who have enrolled in our clinical trial. So that adverse event, thyroid growth, was reversed when we stopped giving FGF1. In our clinical trials, we've injected this into the hearts of patients. It's been in patients with diabetic foot ulcers. We've never seen a serious adverse event to date that was linked to our drug. So we think this drug is relatively safe. And we check in the clinical trial. So for example, in the ALS trial, we'll be looking in the eye to make sure there's no new growth of blood vessels in the retina. We're going to look at tumor markers, make sure there's no bump up in tumor markers in our ALS trial. Again, we've never seen any of these adverse events in humans, only in animals when we severely elevate the dose that these animals get.
Jack, next question. My dad has Bullard palsy, and how would he proceed in being included in the trials?
Yeah, that's it from ALS, although people know that bulbar ALS, that can affect, I think it's about 25% affects, the first affects those ALS patients is in swallowing, where the limb onset ALS affects more of the muscles, but the bulbar palsy would have to be looked at separately in a separate clinical trial for that specific indication. But again, I think that that is a neuron disease, a degenerative neuron disease, and certainly it'd be something we would put on our list if we see activity in our ALS trial and Parkinson's trial that would show that this FGF1 has potential to treat other neurodegenerative diseases.
Hey, Jack, here's a question that you're going to have to answer. May I know the LD50 value of FGF1? I have no idea.
So LD50 is lethal dose, half the lethal dose. So what, and this is the FDA, they make you go up until you basically kill an animal. What is the lethal dose? And that generally, you can establish an LD50 for chemicals, but actually with FGF1, the LD50 is listed as greater than 5,000 micrograms per kilogram. So we couldn't kill the animals basically. So we could cause side effects in the animals, but we couldn't kill them. And that sometimes is seen with natural protein growth factors such as FGF1. So that is the answer to that question.
Okay, now I have another one here that I'm going to try and answer. Somebody asked, would we accept people from Europe for our trial? And I'd like to answer this to you. And I'm going to use Parkinson's disease as an example. When we were told we would have a year delay in Parkinson's disease, we went and filed an application in Mexico, which Jack has informed you. We've been approved to proceed. And as soon as the COVID-19 restrictions in Mexico are lifted, we will start dosing people. In the meantime, people came to us from Estonia and told us they could approve a clinical trial and we might be able to get going in Estonia on Parkinson's disease. We have filed an application. We are waiting for a response in Estonia. Last night, Dr. Jacobs and I were on the phone to a group of neurologists in Morocco who are preparing the final application to file for Parkinson's in Morocco. Now, please understand, we have an application at the US FDA. We are proceeding with the US FDA. But whether it's Mexico, Estonia, or Morocco, if we can find out that the molecule is safe or is not safe, that's valuable information for us. And if we find out it works, and it works in Mexicans or Estonians or Moroccans, and I don't care as long as they're human, then that is valuable data to us. Would we do the same with ALS? Yes. We will go anywhere to initiate trials and get going. And if somebody is in Europe and they meet the criteria, we'll fly them to Mexico. Jack showed you in the heart trial, the woman Constance Donnelly. Every single person in that heart trial cost us $100,000. Every person in the Parkinson's trial is going to cost us $50,000. Are we prepared to fly somebody and put them up if necessary to accelerate the timetable? The answer is yes. But the point being is we are making this aware to you. We are reaching out to you so that you understand what we're doing. If you sign up for our update, if you come to our website and you signed up, you'll get a monthly update. That'll tell you what we're doing and where we're doing it. We are constantly evolving. We have 19 indications. We are learning things. We are expanding the horizons, and we are trying to get clinical trials going everywhere. We've got money. We're looking for results. So, Jack, we got another one in here. Would FGF work on PSP?
Possibly.
Tell me what PSP is first, Jack.
PSP is it's a neurodegenerative disease in the brain. I think any disease that results in a loss of neurons is fair game for us. And I mean, there are hundreds of them, so we obviously can't work on all 100.
Let me take an overview, Jack. Ladies and gentlemen, when we infuse the molecule and it clears the blood-brain barrier in Parkinson's or in ALS or in Alzheimer's, the molecule doesn't have a targeting mechanism. The molecule is in the brain like rain on a field. And wherever the molecules get snagged by the uplifted receptors, it can trigger the process of angiogenesis. Our intended target may be the substantia nigra, or it may be the motor neurons. But once the molecule gets in there and it snags onto the ischemic cells, it can trigger a foci, a growth of new blood vessels, in areas that were not our intent, but where circulation and perfusion are desperately needed. So when Jack says to you, there are many brain diseases, you have to understand, it is my belief. And Jack gets angry with me because I speculate about things and he says, that's easy. He has to prove it. We believe many of these diseases are vascular dysfunction in the brain. The question is a location. If it's in the memory area, if it's in the substantia nigra, if it's in the base of the brain, all of these result in something that the doctors call a different disease. But to us, it's the same thing. It's a drought, insufficient blood getting to where it needs to be. So we believe, as Jack stated, once we start getting permission, many of these diseases are all the same thing. That the reason you have this disease or that disease, or the reason there's 20 different types of Parkinson's disease that have different symptoms, whatever, it depends on where the pipes are broken or clogged. And if we can fix the pipes, then we fix the pipes. So we welcome you to follow us. Once again, you can go to our website and you can sign up for our monthly newsletters. If you want specific information, email me. We've passed our time limit, Jack, so we're going to have to cut this short. Okay. We want to thank you all for coming. Once again, if you can go to our website, which is zgm.care. If you have any questions, you can contact me at dan333@aol.com. Once again, this is a journey of discovery. We do not have answers. We're still struggling to figure out what the questions are. We believe vascular disruption is responsible or a major contributor to these neurodegenerative diseases. As simple as that. We've had doctors who fought us on the heart. We had doctors who fought us on the diabetic foot ulcers who said, oh, no, you can't do this. Well, we've done it. And the point right now is very simple. In the brain, we're either right or wrong. And we want to know that answer as much as you. Now, there's another difference between us. Jack and I have met the people suffering from these diseases. We've met them. We care about them. Jack and I are not worried about our vacation. Jack and I are not worried about being harassed. Jack and I want to get it done because time is not your friend. And we know that. Ladies and gentlemen, Dr. Jacobs and I thank you for your participation. We pray that your struggling is diminished as much as possible. We hope we can make a difference in the long run. We thank you and we bless you. We pray for you. God bless you all. Bye-bye.